So let's talk about the fate of pyruvic acid. First, before I get into that, let's just remember where pyruvic acid comes from. Pyruvic acid is formed from the stuff we talked about last time, what we call glycolysis, which is what's illustrated here on this page. Glycolysis is where we start with that 6-carbon molecule of glucose. We insert a couple energy ATP molecules. There's our 2 quarters going into the slot machine analogy. We pull the lever, we end up getting 4 quarters coming out, but only a net of 2 is actually what we come out with. We oxidize each of those 3-carbon molecules, snagging some of the electrons and hydrogens from it, forming NADH plus H plus or NADH2, however you want to write it. What we're left with are 2 3-carbon molecules, each of which is known as pyruvic acid, or sometimes you'll see it listed as pyruvate, which is not exactly the 100% same thing, but it's fairly close, and they kind of get used interchangeably. What happens to this pyruvic acid and this pyruvic acid after glycolysis is dependent upon what's the state of the cell, okay? So whether or not essentially there is oxygen available inside the cell, we're either gonna, if oxygen is present, carry it into the mitochondria. We call this the aerobic pathway. Remember the term aerobic we've talked about since the beginning of the semester, means with oxygen. This is kind of our preferred mechanism and preferred pathway, because this is where we're gonna end up forming a whole bunch of ATP through what's called the electron transport chain. Before we get to that point, we throw it through what we call the Krebs cycle. And then we get to the electron transport chain. The final stage is the most rewarding as far as the amount of ATP that's produced. Okay? If oxygen is scarce, okay, it doesn't mean that oxygen is completely not in the cell, but we just don't have enough of it, what we'd call anaerobic conditions. We're going to run it through an anaerobic pathway, which results in 2 molecules of lactic acid being formed. And if we think back to when we talked about our muscle lecture, we discussed a little bit about ATP production in muscles, and one of the things we talked about was lactic acid, which I always kind of talk about how it kind of gets a bad rap. A lot of times people like to blame lactic acid for, muscle soreness and being stiff and things like causing cramps and whatnot. It really has nothing to do with any of those things, but it does affect us. And we talked about how it can cause fatigue, for example. Now let's talk a little bit more about this anaerobic pathway. We'll just spend 1 slide on it, and then we'll move on into the aerobic pathway. So once again, this is anaerobic. So this is what we do if oxygen is not present in our cells in a satisfactory amount. We call these anaerobic conditions. This can happen like when your muscle cells are being really, really stressed during really, really strenuous exercise. And that involves the pyruvic acids that we form during glycolysis end up becoming 2 molecules of lactic acid. Okay. Now, if I were to draw a picture, or if I were to draw a line in this picture, like right here on the right side of the pyruvic acid molecules, so right there to the left, this is all glycolysis, where we're starting with that 6-carbon glucose molecule. We're forming a net of 2 ATP. We're oxidizing it, meaning we're taking some of its electrons and hydrogens. What we're going to form as a result is 2 pyruvic acid molecules. If I drew a line on this side and basically bracket it out from here over, from the pyruvic acids to lactic acid, this would be what we call fermentation. And this is the anaerobic pathway, where essentially we do the opposite of what we did back here. We reduce the pyruvic acids, okay? Remember, reduction or reducing means to gain electrons. It's also going to gain the hydrogens with it. Electrons are transferred along with those hydrogens from the NADHs to the pyruvic acids, and in doing so, we form lactic acid. Okay. Now, why are we doing this? Do you see any ATP being formed here? The answer to that should be no. Okay. But what it does, and this is the key right here, we're going to reopen NAD+, meaning that it's going to be able to come and grab some electrons and hydrogens, which is why there's some dotted lines going back in an arrow, feeding back to glucose. Now, we can't run glucose through glycolysis and then turn around and run it through glycolysis again. But you have to remember, there's not just going to be one glucose molecule in a cell. Probably, I don't even know a good number to describe it. It's just a bunch, okay? In order for us to go through glycolysis with any glucose, we need to oxidize, meaning that we must steal its electrons and hydrogen. So we have to have some of these guys available. Okay, these guys are already full. They can't take any more electrons or hydrogens. So we give those electrons and hydrogens to the pyruvic acids. That's what forms the lactic acid. That way we have an empty vehicle to come back, and a new glucose molecule can get oxidized again. So this is what allows for glycolysis to keep going, which allows for a net of 2 ATP to be formed each time we run it through glycolysis. Per glucose molecule, we get 2 ATP. Is this a long-term solution? Absolutely not. Is it going to be super rewarding? Not really either. Okay, because 2 ATP as a net is not a whole bunch, but it is enough that can allow us, to continue to power our muscles as they need that energy as they're contracting, in this case, very strenuously, since we're running through, extraneous exercise. Example. Okay, so the whole thing I would kind of focus on would be this bullet. Why we're doing this, or what's happening. Pyruvic acid is reduced by NADH2 or NADH plus H+, producing NAD+, which keeps glycolysis going. And keeps glycolysis really, might help of new glucose molecules going, if you want to think of it that way. The lactic acids that are formed will rapidly diffuse out of the cell, enter our blood, which can change our pH, because as you know, lactic acids are acids, and acids will make our pH go down in the blood. Thankfully, our hepatocytes or liver cells can convert it back to pyruvic acid, hopefully fairly easily. One thing, once again, I mentioned that lactic acid actually does for us is it does cause muscle fatigue. And I really shouldn't say for us, but does to us. It's not going to contribute to cramps and muscle aches and that kind of stuff like people seem to think. So this is kind of, like I said, like a last ditch effort to keep our muscles contracting, keep that energy providing for the contraction process, but it's not a permanent long-term solution. What we want is to take those pyruvic acid molecules, bring them into the mitochondria, which means that we can continue the process of aerobic cellular respiration. Aerobic, once again, means that we have and use oxygen in this process. The next stage of aerobic cell respiration, I always call it the transition reaction. Some textbooks, I think our textbook just calls it the formation of what we call acetyl-CoA, which is a molecule that is able to enter the Krebs cycle because pyruvic acid is not. Okay. We go through 3 things to get it ready for entering the Krebs cycle. First thing we do to it is we decarboxylate it, which is just a fancy word to describe remove a carbon dioxide molecule from it. CO2 has 1 carbon in it, so we go from 3 in the pyruvic acid. Now when we have 2, it's in the form of what we call acetic acid at this point. This carbon dioxide, along with some carbon dioxide that we form in the Krebs cycle, are the carbon dioxide that we exhale out every time we breathe and exhale. Okay, so that CO2 is formed from metabolism, and we got to get it out of our body. We oxidize it. Oxidize, remember, means to snag electrons. There's the 2 electrons forming NADH plus H plus, or NADH2 if you write it that way. The second hydrogen is not really listed here, but it's there. And then we slap a coenzyme A to it, forming acetyl-CoA. The CoA, short for that coenzyme A. This is what's capable of entering the Krebs cycle, okay? We call this the transition reaction because it transitions us from glycolysis to the Krebs cycle. A lot of textbooks actually kind of just include it as part of the Krebs cycle. In fact, this figure where we're talking Krebs cycle, here is that transition reaction actually just shown as part of it. Not necessarily exactly part of it, but right before it. Both the Krebs cycle and the transition reaction take place in the matrix of our mitochondria. Remember, the matrix of our mitochondria will be the fluid inside of the mitochondria. Our mitochondria have two membranes, an outer membrane and inner membrane, which is folded inward on itself. Inside of that inner membrane is this fluid called matrix, and this is where this takes place. No ATP is formed in this process. Now, if I really kind of want to at some point, I should actually really take this picture and copy and paste it a second time onto this page. Because if I go back to glycolysis, this picture shows not only do we have one pyruvic acid, but we have two pyruvic acids from that original glucose molecule. So really, this should be shown twice because we're only showing one pyruvic acid here. So the transition reaction takes place twice after glycolysis takes place because we have two pyruvic acids. Does that make sense? Okay. Same thing will be seen here for the Krebs cycle. Krebs cycle is kind of the term I use. Some textbooks use citric acid cycle. There's another term, TCA cycle. It's another aerobic stage occurring, like I said, the same place that the transition reaction occurs, matrix of the mitochondria. It's a series of reactions in which a molecule of acetyl-CoA is taken apart. Its carbons are converted to carbon dioxide. Let's just look at it here on the next page, okay? So here's our transition reaction, like I said, where we take our pyruvic acid, we convert it to acetyl-CoA, which is able to enter into the Krebs cycle. We call it a Krebs cycle because we start here and we'll end up back here at the same spot we started with. Now, what's not shown in here, and it's a pretty simplified version of a actual picture that you could show. There's a 4-carbon molecule that's going to be in this stage right here that combines with the 2-carbon molecule that is found in acetyl-CoA. 2 plus 4 is 6. The CoA that was attached to the acetyl-CoA is removed, and what we're left with is a 6-carbon molecule called citric acid. So this is where the term citric acid cycle comes from, okay? We're not going to end up with 6 carbons because, as you can see, we remove a couple of them along with some oxygens forming carbon dioxide. Carbon dioxide, yes, just like what we form back here is what we breathe off. This is where we do a lot of oxidizing of the original acetyl-CoA. We remove electrons and hydrogens, 3 different points using NAD, forming NADH plus H plus. We oxidize using a new coenzyme called FAD, forming FADH2. All of those electrons and hydrogens are going to get carried to the electron transport chain, which are labeled here, just like the electrons and hydrogens that were snagged back here during the transition reaction and the ones that were taken during glycolysis. We do form a total of 1 ATP per revolution, which is not a lot, but it is, a little bit of energy. But looking over here on the the left side of the page, for our outputs, we list 2 ATP. We only show 1. We show 4 carbon dioxides. There's only 2 shown. There's 6 NADH2s shown. We only show 3 in the figure, and then there's 2 FADH2s, but there's only 1 shown over here. That's again because, remember, we have to keep in mind, we had 2 pyruvic acids at the end of glycolysis. So that means we have 2 acetyl-CoAs, which means this cycle takes 2 revolutions. Okay. Does that make sense? Going back to the previous page, let me just read through these last few bullets, and I'll pull that video back up. First carbon is removed by an enzyme that contains vitamin thiamine, leaving a 2-carbon molecule, continues to move through the cycle. 2 more carbons are removed and more pairs of hydrogens are picked up by NAD plus and FAD, another carrier molecule that we talked about the other day. Those hydrogens, and it really should say electrons in there as well, will be carried to the electron transport chain. A small amount of energy is released, enough to synthesize 1 molecule of ATP per turn of the cycle, or 2 turns per glucose molecule, because we get 2 turns of the cycle, because there are 2 acetyl-CoAs that entered it. All right, let's pick up with our video that I started the other day. I want to backtrack us just a touch. So we're still here in glycolysis. They're talking about the end of it.
Enzymes and enzyme assistance. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted.
All right, what they're showing right here, they don't call it what I did, call it the transition reaction. And they're going to form acetyl-CoA. So I'll just back it up just a second. So there's our pyruvic acid or pyruvate, as they have it listed here. Each is 3 carbons. It's 2 of them total.
Those oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called the cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain.
Okay, so she's getting ready to talk about the last stage. We'll jump back in with it here in just a second. Now, let's go back and just keep our heads straight on how much ATP we've got so far. So we won't count fermentation because that's the anaerobic pathway. But here's our glycolysis. We learned that we made a net of 2 ATP, a total of 4, but we remember had to use 2 to start the whole process. So we formed 2 in this process. Okay, we also formed 2 of these NADH plus H pluses, okay, or NADH2s as they're listed here in the figure or at the top of the page. Okay, skipping fermentation to the transition reaction, we form no ATP, but we form a NADH plus H plus for each pyruvic acid. So remember there would be 2 pyruvic acids, so we'd have 2 of these. So far we formed a total net of 2 ATP, but we formed 4 of these guys. Okay, then you take we take ourselves into the Krebs cycle. We add 2 more ATP. So now we've got 4 total ATP, which isn't a lot. Not a lot at all. 4 total ATP is not going to power much. Okay, but what we formed a lot of are these guys. Okay. Before this point, we formed 2 in glycolysis, 2 more in the transition reaction, plus the 6 more that are formed here. We formed 10 of these, 2 of these. So that's 12 molecules that are carrying electrons and hydrogens to our final stage, the electron transport chain, which is a series of electron-carrying proteins containing copper or iron found embedded in the inner mitochondrial membrane, what we call the mitochondrial cristae. The pairs of hydrogens that were once part of the glucose molecule are brought to these proteins by the carrier molecules NADH plus H plus and FADH2. There are going to be 10 of these guys, 2 of these guys carrying those electrons and hydrogens. Here's an illustration of the electron transport chain. Looking at the figure, the location is that inner mitochondrial membrane, shown here in the kind of light tan colored area of our mitochondria. Remember, this is the inner membrane that's folded in on itself. Each of these little purple blobby things is a protein that is part of the chain. It's called an electron transport chain because literally what we do is we transport electron from protein to protein. Eventually, the electrons end up going to oxygen, and along with some hydrogens, form that metabolic water as a byproduct that we mentioned earlier in the lecture. Okay. Where do those electrons come from? Well, they're coming from the NADH plus H plus and the FADH2s. Okay, 10 NADH plus H pluses are bringing those electrons and hydrogens here, and 2 FADHs are bringing them here. Okay. While the electrons go from protein to protein, the hydrogens, you can see there's 3 different pumps that pull those hydrogens into what we call the inner membrane space. There's a little gap between the outer and the inner membrane called the inner membrane space, and that's where this is at right here. So as the electrons get pulled down the chain, hydrogens get pulled into that inner membrane space, which creates a really, really high concentration of hydrogens here compared to the matrix cross-set membrane over here. What do we know about concentration gradients? The drive is always towards equilibrium, right? Towards balance. We always want this and this to be equal, right? We're constantly making this really, really high in hydrogen ions, and this is not high in hydrogen ions. But we're going to allow for hydrogen ions to come down their gradient using an enzyme. And the name of the enzyme is absolutely perfect because it's telling you exactly what its job is. To synthase ATP. As those hydrogen ions come through that enzyme, it gives that enzyme the energy that it needs to add that phosphate to each ADP, forming ATP. Okay, so let's read through. Each hydrogen that is brought to the electron transport chain by NADH plus H plus and FADH2 is split into its proton, hydrogen ions, and its electron, E with the minus sign. Electrons are passed from one protein to the next, finally to oxygen. Reactions of the electrons with these proteins release most of the energy contained in that glucose molecule. Each oxygen that is gained to electrons reacts with 2 of the hydrogen ions, forming water. Metabolic water is what we call it, contributes to the necessary intracellular fluid, also prevents acidosis. Because if hydrogen ions accumulated, they would rapidly lower the pH of the cell. The formation of this ATP from the ADP and PI is called oxidative phosphorylation. We call it that because it is an oxygen-requiring process. And look how many ATP we make during this stage alone. 32 to 34 total from that original glucose molecule that we started with. So once again, glycolysis, we netted out 2. Transition reaction, we had 0. Krebs cycle, we had 2. Now look how much we get from that glucose. 32 to 34 of these guys. And it's all because of the electrons getting passed from protein to protein, allowing the hydrogens to fill the space, and then come down their gradient, empowering that enzyme called ATP synthase. This segment of our figure is an illustration of what's called chemiosmosis. So let's talk about that on this page. Actual production of ATP in cellular respiration takes place through a process called chemiosmosis, which involves the pumping of protons. Remember, protons, that's what H plus is. Okay, this is just showing you kind of a very simplified version of the electron transport chain. Okay, all of those purple blobs that were on this page, they've just combined it to kind of one block in this page. The electrons are passed from protein to protein, which enables the hydrogen ions to be carried or pumped into that intermembrane space, establishing a high hydrogen ion concentration here compared to here in the matrix. Okay. This pumping establishes a proton gradient. After gradient established, protons pass down the gradient through the enzyme called ATP synthase. ATP synthase uses the energy of those protons to generate ATP using ADP and phosphate ions. And that's the starting point. Let's finish our video so we can see what's really taking place.
The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain.
That's all right here.
High-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that enter the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide.
Okay, so hopefully the video really kind of emphasized what I was getting at with all of this stuff. Okay, things to make sure you know, really pay attention to things like where these processes are taking place. You know, like the electron transport chain, know that it's in the mitochondrial cristae. I'm not going to ask you to know every single piece of information that I include on these slides, but definitely know, you know, like the inputs, outputs, that's a big thing. When telling me, you know, how many ATP are formed during that stage. In this case, the electron transport chain, you would know 32 to 34 formed during this stage alone. Now, putting it all together, you're also going to want to know how much ATP that we get total from that glucose, which is 36 to 38. And that's what's discussed on this page. So let's kind of summarize it all together and talk about why it's not a solid number, it's a range. For every molecule of NADH plus H plus that drops off hydrogen atoms to the electron transport chain, 2 or 3 molecules of ATP, or an average of 2.5, are produced via oxidative phosphorylation. For every molecule of FADH2 that drops its hydrogens off at the electron transport chain, only 1 or 2 molecules of ATP are formed, which is an average of 1.5. So you might be wondering, why do we get less energy from the FADH2 than we do for the NADH plus H plus? Well, if I go back to this illustration, it is shown right here why. NADH plus H plus, no matter where it's coming from, whether we form this NADH plus H plus in the in the glycolysis stage, transition reaction, or the Krebs cycle, it always dumps its electrons off and hydrogens right here at that first pump. Okay, which means that we start to pull hydrogens into that intermembrane space right off the bat. Whereas with FADH2, it dumps its electrons off kind of more down the chain, the second stop, which means it doesn't get any hydrogens across to down here till we get to this pump here, which means less hydrogens come into this intermembrane space from the FADH2s than they do from the NADH plus H pluses. So that's why we don't get as much ATP from the FADH2s as we do the NADH plus H pluses. Okay? So that's what this states. This difference is due to the fact that FADH2 drops off its hydrogen atoms at a lower energy level, complex 2, than NADH plus H1, H pluses, which is complex 1. So one glucose molecule, when you add everything up from those 4 total stages, I say 3 here, but I didn't include the transition reaction between glycolysis and the Krebs cycle. Really, I should, because it's a stage. And then adding the electron transport chain. That one glucose molecule that we started with yields out a total of 36 to 38 ATP. And you might wonder why it's a 36 to 38, not a solid number. Well, it all kind of is traced back to the NADHs that were formed during glycolysis. Remember, glycolysis takes place out in the cytoplasm, outside of the mitochondria. Everything else, like right here, the transition reaction, the Krebs cycle, electron transport chain, all those take place in mitochondria. So those NADHs that were formed during glycolysis have to get carried into the mitochondria, and in doing so, sometimes it requires a little bit of energy. And I'll just kind of leave it at that. So here's where the variability comes in. Notice here from those 2 NADH plus H pluses, we get 4 or 6 ATP. You add all those up, you get 32 or 34, and I just listed that as 32 to 34 here a couple slides ago. 32 to 34 was just from the electron transport chain. We had 2 from glycolysis, 2 from Krebs. 4 plus 32 is 36. 4 plus 34 is 38. So that's where the total numbers come from. Now, this is only possible if oxygen is available. That's why we call this aerobic respiration. The oxygen isn't really going to play a role until the very end, but we can't go through the electron transport chain without oxygen, and we can't get any energy from anything before that without oxygen. So oxygen is super, super important for making a lot of energy, as it says here. Summary of the 3, I really should say 4 stages of cell respiration, also includes vitamins and minerals that are essential for the process. So it's not only important to get a diet consisting of carbohydrates, and we'll get into proteins and fats here in a second, but it's also important to get a well-balanced diet that includes vitamins and minerals. Like NADH plus H plus, FADH2, and we talked about how vitamins were involved with making those things. There's a lot of other things that go into, making the energy that we need. Vitamins and minerals do other things for us as well. So eating is important. And also, none of this is possible without breathing oxygen in and getting the CO2 out that we produce. Otherwise, we become acidic, as we've talked a little bit about before. Table 25.1 is just a summarization, basically just kind of the same thing as what's shown here in the figure, just list it out. I'm not going to read through it, but I include it because it's your textbook. And then just another illustration showing a summarization, breaking it down step by step. Glycolysis, what's taking place outside the mitochondria. Formation of acetyl-CoA, I call that the transition reaction, Krebs cycle, and then the electron transport chain, and all the different ATPs that are formed along the way. Okay, so that is cellular respiration, where we are taking glucose apart, forming that energy, which is an example of catabolism. Now, thinking back to the title of this lecture, we said this is the cellular metabolism lecture, and that first slide that we really talked about things, we had talked about that slide before. We'd seen that slide back in our chemistry lecture where we discussed that metabolism included two stages or two phases, anabolism and catabolism. We kind of just focused on catabolism of carbohydrates, breaking things down from bigger to smaller and releasing useful energy from it. As you can see on this page, we've got the slide title, Carbohydrate Anabolism. So that would be the opposite of catabolism, building up from smaller to bigger. Even though most of the glucose in our body is catabolized to generate ATP, glucose may take part in or be formed via several anabolic reactions. If it's not necessary for ATP production at that point, we can take glucose molecules bonded together, forming glycogen. We call this glycogenesis. Glycogen is a polysaccharide form of glucose. Stimulation is due to the hormone insulin that we release when we are hyperglycemic. What does hyperglycemia mean again? High blood sugar, high blood glucose. We store glycogen in places like our hepatocytes or liver cells and our skeletal muscle. And this is what you do if you're an athlete when you carb load. We talked about that previously. Thinking back to earlier in the lecture, I'll just type them up real quick. We had those 6 buzzwords. The first word we said was glycolysis, which we just got done talking about, the breakdown of glucose to form ATP molecules. The second term, one of the second terms, I don't remember which exact order we went. Glycogenesis, this is the term that we just mentioned, is the formation of glycogen from glucose molecules. Opposite of glycogenesis is glycogenolysis, which is the breakdown of glycogen into glucose molecules. And then there's gluconeogenesis, which is the formation of glucose molecules, non-carbohydrate sources, things like lipids and proteins. We'll add to our list here more in a second. Okay. Now, we do glycogenesis as a way to store up our glucose in high quantities. That way, when we need it, we can call on it. Okay. Now, the title of this slide, it says carbohydrate anabolism, but this bullet here and this bullet here is actually an example of carbohydrate catabolism. Okay, there's more than just glycolysis and what we just talked about with the other parts of cell metabolism or cellular respiration that are catabolic in nature. Glycogenolysis is another catabolic reaction involving carbs. So like I said, when athlete carbo loads before a race, they store up that glycogen. That way during the race, they can call on that glycogen to make ATP. Glycogen stored in our liver and muscle cells can be broken down into glucose to be released into the blood for uptake by cells, and then we go through cell respiration. So going through glycolysis, transition reaction, Krebs cycle, electron transport chain stuff that we just talked about. We call this process of splitting glycogen into glucose glycogenolysis, stimulated by the hormones glucagon. Remember, I always told you guys to think glucagon like glucose is gone, which is just another way of saying hypoglycemia. The opposite of hyperglycemia, hypoglycemia, therefore, is low blood sugar. And then epinephrine is another hormone. We know that's released from our adrenal medulla whenever we have that fight or flight response activated, which, you know, should make sense. We are going to release glucose in the blood when we're in a fight or flight response, because glucose can then be used to make ATP through all those things that we just talked about, which gives us the energy that we need to fight or flight. Glucose can also be formed from triglycerides or fats and proteins in response to low liver glycogen or a fasted state. The glycerol part of a triglyceride, which would be a 3-carbon molecule, lactic acid, which we just talked about, is a 3-carbon molecule, certain amino acids. We can basically just kind of add those things together and form in our liver glucose from those non-carbohydrate sources. We call that gluconeogenesis, the fourth term I just typed up. Stimulated by the hormone cortisol, which we nicknamed earlier, talked about earlier this semester, is our stress hormones released by the adrenal cortex. And we need energy to combat that stress. Glucagon and the T3, T4 thyroid hormones are other important hormones that promote gluconeogenesis. So illustrations of those different terms are shown here on this page. On the left, we show, I'm going to kind of simplify the figure, going from blood glucose to glycogen with these red arrows in that direction. We have what's called glycogenesis taking place in the hormone insulin involved, going from glycogen to glucose, breaking it down, in other words. We are going through glycogenolysis using glucagon and epinephrine. Could have got into much greater detail, as you can see, what's taking place, the enzymes involved, but I'm not going to do that. It's not, you know, something I'm going to cover in this class. Over here on the right, we're showing you how we can make glucose from things like amino acids and lactic acid, as well as from the glycerol of a triglyceride. So from non-carbohydrate sources, which is an example of gluconeogenesis, stimulated by cortisol glucagon, and it doesn't list it for whatever reason, but the T3, T4 thyroid hormones as well. Okay, that's it for carbs. But there are 3 fuel nutrients that our body ingests. Proteins are a second fuel nutrient. Not a prominent, super important fuel nutrient. We kind of prefer to use carbs and fats, but we can use, when needed, proteins as an energy source. Thinking back to our biochemistry lecture at the beginning of the semester, we learned proteins are made up of smaller molecules, building blocks known as amino acids. Here's an illustration of an amino acid. Okay. The primary source and use for our, not primary source, but the primary use for amino acids we obtain from our food is just to synthesize new proteins, like the proteins that are in our muscles or proteins anywhere else that are going to give us, you know, a lot of different functions. Remember, something's happening in the body, we can almost guarantee it's a protein doing it. When we have excess amino acids, in other words, those that are not immediately needed for protein synthesis, we can use them for energy production. But we have to go through a process called deamination. where we remove the amino group from that molecule. So the NH2 section of the amino acid is removed, which occurs in the liver. Okay. In doing that process, we form what's called ammonia, NH3, which is very toxic to our body. But thankfully, we're performing this in our liver. In our liver, before that ammonia can leave and enter our blood in high quantities, we convert that ammonia to urea, which is much less toxic and is something that we easily eliminate out of our urine. In fact, it's a very prominent component of our urine that we are getting out every time we pee. Okay. But what's really not important is this stuff for the ATP production aspect of things. Going back to this molecule over here where amino acid is, when we remove that NH2, essentially what we're going to be left with is a molecule with some carbon in it. A couple carbons like we show here, and a couple more and a couple more. You add those up together, you can get the components that you need to fit the Krebs cycle. You know, thinking back to the Krebs cycle, we learned that acetyl-CoA is what entered the Krebs cycle, right? Acetyl-CoA was a molecule with 2 carbons. Well, here we have 2 carbons. Okay, we can take those amino acids, attach the coenzyme A to it. Basically, you've got the same thing as what you had after the transition reaction with our pyruvic acids, and we can enter that into the Krebs cycle, no big deal. Okay. Not what we would really prefer to do, but it's something we can do. What we would prefer to use if we can't use carbohydrates are fats. Fats or lipids like carbohydrates can be catabolized to produce ATP. If our body has no immediate need to use the lipids in this way, we store them in our adipose tissue throughout our body and our liver. Unfortunately, humans have an unlimited capacity to store fat in our body, and a lot of Americans choose to take advantage of that, and that contributes to our current health issues and what we call the obesity epidemic, not something I'm going to talk about in great detail. When we use those triglycerides for energy, we use an enzyme called a lipase enzyme that hydrolyzes or breaks it into its components. A triglyceride is illustrated down here at the bottom. It's made up of kind of 4 things. A 3-carbon, what we call glycerol molecule, and then 1, 2, 3, what we call fatty acid chains, fatty acid molecules attached to that glycerol. Basically, we split all these things apart. The glycerol is pretty darn similar to pyruvic acid. Both glycerol is 3 carbons, so is pyruvic acid. So we can take that glycerol and basically convert it to pyruvic acid and then enter it into the Krebs cycle once we've converted it to acetyl-CoA. It's not going to provide nearly as much energy as what we get from those fatty acid tails. Those fatty acids are catabolized differently. Notice there's a whole bunch of carbons, and there's a whole bunch of hydrogens. Actually, there's more hydrogens. But more importantly are the carbons to kind of focus on right now. What we do is a process called beta oxidation, where we come in and we just snip off 2 carbon segments at a time. And then those 2 carbons, you slap a coenzyme A in it, you've got acetyl-CoA essentially that can enter the Krebs cycle. And each of these fatty acid tails can contain like 20 carbons, which means you can get 10 different 2-carbon molecules, 10 more here, 10 more here. You can get a whole bunch of things that can enter the Krebs cycle, which means that we can get a whole bunch of energy. Okay. So let me read this. Since fatty acid chains can be around 20 carbons in length, 10 acetyl-CoA molecules or so can be formed entering the Krebs cycle, providing enough energy molecules or enough molecules to yield 100 ATP. So we can get a whole bunch of energy from one molecule of a triglyceride compared to the 36 or 38 from that just singular glucose. This explains why things that are fatty, like in our diet, are much more high in calories. A calorie is just a measurement of how much energy you get from a food substance. If you compare a gram of fat and a gram of carbohydrates, a singular gram of fat gives you 9 calories of energy. Whereas, and this is the same for proteins, but carbohydrates, both proteins and carbohydrates, per gram only give you 4. So it's over double the amount of energy from fats than it is from carbohydrates and proteins as well. And we talked about that back at the beginning of the semester when we first kind of introduced fats, but wanted to kind of reemphasize this. Now, all this stuff is what we call lipolysis. Lipolysis is breakdown of fats or triglycerides. And it's just like what we do with glycolysis to form ATP. Opposite of that is lipogenesis. Like I said, we have an unlimited ability to do this, which is the formation of fats or triglycerides in our body. It doesn't necessarily have to be where we take fats in our body as in, eating fats in our diet. We can make fats from excess carbs and proteins as well, which explains why if you eat candy all day, even though it's not fatty, it's really sugary. Those carbohydrate sugars can be converted to fats in our body. And that's what happens. Now, as part of normal fatty acid catabolism, as part of normal fatty acid breakdown, our liver converts some acetyl-CoA molecules into substances called ketone bodies. This includes acetoacetic acid, beta-hydroxybutyric acid, and acetone. We call the formation of these 3 substances ketogenesis. Once they're produced, ketone bodies leave our liver, entering our body cells where they're broken down into acetyl-CoA to enter the Krebs cycle. The level of ketone bodies in our body is normally very low because other tissues use them for ATP production as fast as they're being formed. During periods of excess beta oxidation, however, production of ketone bodies can exceed their uptake and use by our body cells. So we make more than we can use, essentially. This happens like after you eat something really rich in triglycerides, when you're in a fasting or starvation state, because there's not many carbs available for catabolism. Excess beta oxidation may also occur in poorly controlled or untreated diabetes mellitus, diabetes for short. When the concentration of ketone bodies in our blood rises above normal, we call this condition ketosis. And those ketone bodies, most of which are acids, must be buffered. And if not, our pH will drop. We call this condition ketoacidosis. And our decreased blood pH will cause things like depression of our central nervous system, resulting in disorientation, coma, and even death if not treated. When a diabetic becomes severely insulin deficient, one of the telltale signs is the sweet smell of their breath from the ketone body acetone. And they even say that their urine will smell really fruity. It's kind of crazy, but that's what happens when somebody's not doing a good job regulating their insulin and blood sugar levels. Now, you've probably heard it's very kind of a forefront fad right now, keto diets, right? Probably heard of that. People are like, not eating many, it's kind of just a different way of saying like a low carb diet. It's a little bit different, but basically they're trying to promote a lot of this stuff where your body's not containing many carbohydrates So what you do is you burn fat instead. You know, it can be very beneficial and cause a lot of weight loss for sure, definitely. As long as you're, you know, being careful and you're not screwing with your blood sugar too much as a result of it, you know, it can be a pretty successful way of losing some weight, losing that bad fat that we like to store up. Illustration here on the next page just shows, you know, I talked about how we can go from amino acids, eventually go to the Krebs cycle. Same thing for triglycerides. We can use the glycerol to get it basically converted to pyruvic acid, then eventually get to the Krebs cycle. And then through beta oxidation, take those fatty acids apart, 2 carbon by 2 carbon by 2 carbon, attach a coenzyme A to it to enter the Krebs cycle and get a whole bunch of energy from it. In doing so, like I just said, we can form ketones. Over on the right just shows like different entry points where you can plug things in as part of the cellular respiration process we've talked about. So that is a finishing of our metabolism aspect of things. We talked first about carbs, both catabolism and anabolism. Talked a lot about catabolism of glucose through the various aspects of ATP production. And we talked just a little bit about making glycogen, making glucose from non-carb sources. Literally just talked about catabolism, anabolism of triglycerides. What we really didn't talk about on this entire table was this very last thing, protein anabolism. But the reason is we already have talked about it. Thinking back to the very beginning of the semester, when we talked about what we call gene expression. Remember, gene expression was a process where we take a DNA template inside our nucleus, we transcribe it, forming RNA. We take that RNA and using ribosomes, our protein factories, we take amino acids and we put them together, forming proteins. So going from DNA to RNA to proteins, I've written that kind of formula out many times this semester. That's just another way of saying protein anabolism. Something that we're doing a lot of all the time. Something that's super important. But I won't get into it in any more detail because we've already really kind of talked about it. Which leaves us with one final thing to talk about. Regulating our body temperature, or thermoregulation. It's kind of a fancier way of saying it. And this all relates, and it's the reason why I included it as part of this lecture, to metabolism. Because thinking back to the very beginning of the lecture, we said whenever any metabolic reaction takes place, whether it's catabolic or anabolic, heat is produced. Okay, and you're looking at this illustration. Catabolic reactions release heat. Anabolic reactions release heat. This heat that we produce, or as it says, release, gives us a body temperature. So let me read through. Your body produces more or less heat depending on the rate of its metabolic reaction. Because homeostasis body temperature can be maintained only if the rate of heat loss from body equals the rate of heat production by metabolism, it is important to understand the ways in which heat can be lost, gained, or conserved. So what is heat? It's a form of energy. It's kinetic energy that can be measured as a temperature. And despite wide fluctuations in environmental temperatures, things that we have experienced, you know, in these couple months of class, going from when it was like 100 degrees to now, the wind chill's like 30 degrees. We have homeostatic mechanisms that can maintain a normal range of internal body temperature. We keep a pretty darn consistent body temperature around 37 degrees Celsius, even though we're constantly being exposed to extreme cold, extreme heats, that kind of stuff. If the rate of body heat production equals the rate of heat loss, our body maintains that constant core temperature, like I said, around 37 degrees Celsius or 98.6 degrees Fahrenheit. Since heat is a product of metabolism, it's important to understand metabolism. We should have a great understanding of metabolism now. We can measure our rate of metabolism. We call this our basal metabolic rate. It's the measurement of an individual's metabolic rate with the body in a quiet, resting, and fasting condition. And that's important because that's basically a direct indicator of how much heat we're producing. Two different forms of temperature can be taken of a person's body, their core and their shell. Core temperature is the temperature of their body structure deep to the skin and subcutaneous layer. So it's in their core, hence the name. That's usually what we see around 37 degrees Celsius and 8.6 degrees Fahrenheit. If that temperature is too high, it kills our body by causing the proteins in our body to denature. Remember, they are like a slinky, they can lose shape, which causes them to lose function. Core temperatures too low causes cardiac arrhythmias. In other words, the heart loses kind of normal rhythm. Then that can result in body shutting down and dying. Shell temperature is the temperature near the body surface. In other words, in our skin and subcutaneous layer. This is what's usually measured. Like when you go to the doctor or like anywhere you go right now, like I went to the courthouse the other day to, remove my tags. I had to, scan my wrist before I could go in. They're taking my temperature on my external surface. Truly, it's not always a great indicator of what my actual temperature of my body is. Because if I was standing outside without a jacket on for 5 minutes before I did that, my body temperature and my skin temperature, my shell temperature would probably be pretty low compared to my more core temperature. It's done though because it's a lot simpler. To take a core temperature, you actually have to insert stuff into the body, which means you're gonna like stick a probe down or up an orifice. Okay, like a rectal temperature, you can probably guess what that is. Going up the rectum, esophageal. Another way to do it, you go actually up through the nose and down into the esophagus. You walk to the courthouse and you want to go inside and pay your taxes. They tell you, okay, we got to stick a thermometer up your butt. You're probably not going to do it, right? Okay. They can scan your forehead, scan your wrist, whatever. But just know a shell temperature, it says it can be 1 to 6 degrees lower, but it could also be higher. Because if you're like exposed to the sun when it's really hot outside, it might be warmer than your internal body as well. So keep that in mind. As a tissue increases its metabolism, the amount of heat it produces also increases. So any increase in metabolism increases heat production. So it's a nice, not inverse relationship, but it's just a straight relationship. One goes up, the other goes up. Several factors influence your metabolic rate, which therefore affects the amount of heat you're producing. Exercise, obviously exercise is going to make your metabolic rate go up, therefore heat goes up. Thyroid hormones, that's all they do, that's their main job, is to increase metabolic rates. So they make your body temperature go up. Same thing for epi and norepi. Increase in body temperature affects metabolism, which affects your heat production. Ingestion of food, age, gender, climate, sleep, and malnutrition are other things that affect your metabolism, which affect your heat production. Once again, maintaining normal body temperature is something that we must do, and in doing so, we must match the amount of heat we produce with the amount of heat that we are dissipating or losing to the environment. Now, this semester we've talked a lot about gradients. We were just talking about like hydrogen ions and how they go down the concentration gradients. Recently, we talked about pressure gradients and how blood flows, how our air moves in and out of the lungs. Well, temperature moves down gradients as well. And we call these temperature gradients. And this is something that, affects, like if you leave your door open, when it's hot outside, you're going to actually let heat in. So when you leave the door open, somebody says shut the door, you're letting all the cold air out. You're actually letting the hot air in. Okay? The heat's coming in from high to low. Okay? Our body can transfer heat to the environment 4 ways: conduction, convection, radiation, evaporation. Okay? Conduction is where heat exchange occurs between molecules of 2 materials, and the key here is that they're in direct contact with one another, like the cold surfboard in his arms. Like you guys sitting in those cold desks. This room's temperature is set, the thermostat's probably around 70. That means when you come in here, the desks are at 70 degrees. 70 degrees Fahrenheit is quite a bit colder than 98.6 degrees Fahrenheit. So putting your arms or your legs up against that table, you're going to transfer that heat from your body. You're going to heat the table up, and that table is going to cause you to cool off because it's basically zapping your heat. Sitting in a hot tub would be an example of a way that our body could gain heat. Okay, because typically hot tubs are 100 degrees or so, which is warmer than our body. Okay. Convection is the transfer of heat by the movement of a fluid. Now, a fluid could be like the water that he is standing on in the wet sand, or if he was to jump in that water. But gases, in this case, are considered a fluid as well. So the gas that's in the air, that when it moves across your face, like if you fan your face or if a breeze hits your face, is a way to transfer heat to the environment. Okay. This explains why it feels good when you're hot to fan your face. You're increasing the amount of movement of air across your face, and the heat is going to be dissipated to the atmosphere as a result. Now, sitting in a hot tub is also a form of convection. Like we just said, it's a form of conduction, okay, because water is a fluid. Now, speaking of water and gases, or liquids and gases, any guess as to which, and it's a big difference, has a better ability to transfer heat? Is it a gas like the air, or is it a liquid like water? Which one is able to transfer heat more readily? Air or water?
Air.
It's water. Okay. We can transfer, I think it's like 20 times more efficiently our heat to water than we can to the air. So here's an easy explanation, demonstration, the difference. And walking outside on a day that's like 50 degrees. It's a little chilly, right? If you're outside without a jacket on, you're probably a little bit cold. But your body, should be able to shiver enough that you're going to be okay. Not susceptible to hypothermia at 50 degrees. What if you were to jump in a river, in this case, looks like an ocean, that's 50 degrees? It's a lot colder. 70 degrees is a very pleasant day, right? Swimming in a pool that's 70 degrees is awful. That's like the water temperature when the pool opens, like around Memorial Day. It is cold and it sucks to be in that 70-degree water, but you get outside, if it's 70 degrees, you're going to feel fantastic. It's like perfect temperature outside. That's because the water is zapping your energy, your heat, I should say, a lot more effectively than it would be from your body to the air. Radiation is a transfer of heat in the form of infrared rays between a warmer object and a cooler one without physical contact. A great example would be the radiation providing heat to our body from the sun. And then evaporation is conversion of a liquid to a vapor, which involves both evaporated water from our skin, in other words, from our sweat, and the water exhaled from our lungs. And this is why, like, dry fit Under Armour style clothing is really good at keeping you cool compared to just like a cotton t-shirt, because those types of clothing wick away the water and allow for your sweat to evaporate as you exercise, which is the main defense against overheating during exercise. Unless, of course, you're like exercising in a pool, because then you're, transferring that heat via convection. But like if you're outside for a run, or you're playing soccer, baseball, or whatever, summertime, wear moisture wicking material. Don't wear, obviously, not wool or something like that, but cotton t-shirt that's just soaked with sweat is not really good at keeping your body cool. Okay, finally, let's end our lecture talking about the specifics and what happens in our body and how we control our body temperature. Something that we can trace back to the very beginning of the semester when we first introduced feedback loops. We learned, about a third of the way through the semester that the main area of our brain responsible for regulating our body temperature is our hypothalamus, specifically what we call the preoptic area of our hypothalamus. It receives impulses from thermoreceptors, which are just hot and cold receptors in our skin, as well as the mucous membranes, and even there are thermoreceptors in the hypothalamus itself. As a result of those impulses coming into our hypothalamus will activate two other areas of the hypothalamus, a heat promoting center and a heat losing center. Okay. The heat promoting center is going to promote heat formation in our body. In other words, that means we're going to try to do everything we can to raise our body temperature. So this is what we do when we get cold. And what's the most obvious thing that we've talked about that we do when we get cold? We shiver. And that's just one thing that we do. Shivering does increase body heat production by about 4 times the basal rate in just a few minutes. So it's super effective. But there's also what we call non-shivering thermogenesis, which is where we release some hormones, T3, T4, epi, norepi, because they increase cell metabolism. And as I just said, any increase in metabolism results in an increase in heat production. So these are like non-shivering ways of increasing body temperature. There's also, we talked a little bit about this before, what we do in our skin. We decrease the flow of blood to our skin by way of constricting blood vessels in our skin. This is a sympathetic response. We redistribute that blood flow to our internal organs. It's less likely to result in heat loss to the environment through those different mechanisms that we just talked about here on this page. If your body heat is more internal, it's harder to get it out of the body through conduction, convection, etc. Okay. Our heat losing center is what we do when we are hot. We're trying to cool our body off. What's the most obvious prominent thing that we think of when we are hot? We do what? Sweat. Okay. Sweat, as we just said, is going to help try to promote evaporation, the transfer of that heat when we convert water into a gas. We're also going to decrease metabolic rate any way we can because that decreases heat production. And we'll do the opposite thing that we did when we are trying to conserve heat. We'll dilate our blood vessels. This is why your face a lot of times turns bright red when you're really hot. Your skin is loaded with blood, and that blood is the heat that we're going to try to dissipate to the environment via radiation conduction mechanism that we just talked about on the previous page. Sound good? So there's a lot more that I didn't talk about early in the semester, aside from shivering, aside from sweating, that we do when we're hot and when we're cold. Okay, any questions?